This video will focus on explaining the mechanism of RNA interference, also called RNAi. RNAi is a general term used to refer to the cellular tool that is able to dial down or prevent expressions of a gene through non-coding RNA. RNA interference is done by non-coding RNAs and is often called RNA silencing. It can be done before transcription even occurs, so that the mRNA isn't ever made, and this requires action within the nucleus and on the DNA, or it can be done post-transcriptionally, which means that the mRNA is degraded after it's made and before it can become a protein. siRNA usually uses the second method, and siRNA is the one that this lesson is focusing on. There are, however, three main common types of RNA within the RNAi group. These are short, also called small, interfering RNAs, or siRNA, microRNAs, or miRNAs, and peewee RNA, also called peewee interacting RNAs, or piRNA, named after the special type of Argonaute protein that they interact with. These are unique because they only work in the gametes. In order to create siRNA, the type we're focusing on, the double-stranded precursor must be broken down into smaller pieces by dicer proteins. The precursors and the new small 20 to 25 nucleotide long siRNA pieces have a non-guiding sense strand and an antisense guiding strand that is complementary to the mRNA, which is how it guides. This is often called an RNA duplex. The process begins when precursor double-stranded RNA, this could be viral if it was occurring naturally, or synthetic if we're using it for a medical purpose, is degraded into siRNA by dicer endonuclease enzymes in the RNase 3 domain. The siRNAs, now around 20 or 21 to 25 base pairs in length, have a 2 nucleotide base overhang on both their 3 and 5' ends. And these join an RNA complex called the RISC complex, the RNA-induced silencing complex. This complex includes effector proteins that then act on the complementary RNA and degrade it. The incorporation of effector machinery occurs, which either silences or destroys the mRNA. There are many different types of proteins involved, however they are always a member of the Argonaute protein superfamily. The siRNA is loaded inside the Argonaute protein, often Argo2, and is now able to recognize target RNA through simple Watson and Crick complementary base pairing. And this is done by the antisense strand, and it's quite reliable. It's the sense strand that is removed in the RISC complex, and upon cutting and releasing the sense strand, the RISC complex becomes activated, and it is now able to destroy mRNA pre-translation before it can ever become a protein, and therefore prevent it from having a negative effect